Thank you. I'll now, without further ado, call on Francoise to give her lecture for our symposium. Thank you, Francoise.
Chancellor, Honorable Michael Kirby, dear David, dear distinguished delegates and colleagues and ladies and gentlemen, I would like first to express my very deep thanks to you, Chancellor, to you, David, for this honorary degree. It means a lot for me, for many reasons. As the minister said a few minutes ago, the Kirby Institute and the Pasteur Institute has a lot in common. Both are very much interested in infectious disease. The missions also included trying to strengthen the human capacity, human resources in the developing world. So for me, this honorary degree, it's really making a link between our both institutes, and I really hope that this degree will be a possibility to have more collaboration between our two institutes in the future. Particularly David, and we talked yesterday evening about that, to have collaboration and strengthen collaboration in Cambodia, a country where we have both been very much involved. It means a lot also because I know David since the early 80s, and I know what he has done for the fight against AIDS. AIDS in Australia, AIDS in many parts of the world, and for me it's really wonderful to be here today, and I would like really to thank you, David, for what you have done, what you are still doing, what you will do in the future, and thank you for inviting me to this symposium. My talk today will be on some of the work in my lab, where trying to contribute at least to a better understanding on how to control HIV infection. So what I like to do today is to go through some of this work, which is trying to understand better, particularly how to control two main determinants of HIV pathogenesis, which are summarized in this slide. Mainly, of course, HIV infection itself, the establishment of HIV reservoir, which we know today are critical in the evolution of the disease. And the second very important determinant, which indeed are related to each other, the fact that this infection is capable to induce abnormal inflammation, abnormal activation, immune activation. Both are linked, and we know that if we want to control probably this infection, we have to control these two critical determinants of HIV infection outcome. So my lab is trying really to bring some contribution in that area by studying different models of protection against the evolution of HIV or SIV infection. One, models to try to control HIV infection itself, or models to try to control abnormal immune activation. Both models are in human and non-human primates. The first model that I'm going to talk about is the work Azier Syrian in my lab is doing on a lead controller, and the second type of work he is doing more recently on post-treatment controllers on a cohort of patients that we have now in France. So let's start by the lead controller. Azier and his team and several others, like Bruce Walker, have reported that in a lead controller there is a very efficient CD8 response, suppressive CD8 response, which is capable to control, at least to suppress HIV infection. This is summarized on this slide, showing here, and this is the work of Azier, who really add non-stimulated CD8 cells to CD4 cells of elite controllers after infection by HIV, and as you can see on this slide, nicely showed a very nice control of HIV infection with this non-stimulated CD8 cell from the controllers. So, in order to understand better the function of these cells, first of all, he showed that this strong CD8 T cell capacity was found in elite controllers, but was not present during primary HIV infection in HIV patients. It's a study on 50 patients reported here, and you can see on this slide the differences in the capacity of the CD8 cells to suppress HIV infection in the elite controllers compared to patients during primary HIV infection. So the hypothesis is that probably this very high suppressive capacity of the CD8 T cell of the elite controllers is probably related to intrinsic characteristic of these cells, in particular, as reported on this slide, their capacity to kill the CD4 infected cells, their capacity to produce cytokine, and their proliferative capacity as well. And the most hypothesis is that indeed we have proliferation in the elite controller of these cells with those characteristics which are not present in the progressors. It has been also reported in the literature the high functional avidity of these cells, as well as MHC and TCR plasticities that I like to mention here. More recently, we have been able to find out a particular phenotype of the cells with this capacity, as shown on this slide. The CD8 suppressive capacity is mostly related to cells that are expressing HLA-DR, but are CD38 positive, negative cells, as shown in black on this slide. So there is clearly an expansion and probably a proliferation of cells that carry this HLA-DR positive, CD38 negative phenotype, and which are probably responsible for this effector function. We started to wonder whether this is related to the priming of the CD8 cells by a monocyte-derived dendritic cells. And we started to characterize the MDC of elite controllers, and this slide is showing the transcriptional profiles that we observe by microarrays, indicating, as shown here, a non-inflammatory profiles in the MDC of the elite controllers. So we started to study a little bit in more details the MDCs of the elite controllers. We reported that the MDC are resistant to HIV infection, as shown here on this slide, compared to the cells of healthy donor. More recently, we look whether the MDCs derived from the monocyte of elite controllers were capable to capture HIV, and you can see here that they have a high capacity to capture HIV, which is shown on this slide here. So we can say that probably MDCs from elite controllers, from HIV controllers, are playing a role in the priming of CD8 T cells in the controllers. The MDC, first of all, have this reduced susceptibility to HIV infections that might play a role in preserving the function of the MDC. The fact that the MDCs have an enhanced capacity to capture HIV may also play a role in the efficient induction of CD8 T cell response, despite a very low antigen level in the controllers. We also found out these non-inflammatory transcriptional profiles in the MDCs, that this may be related also to the low inflammatory context of the elite controllers. So when we look over time in the controllers, in fact, the CD8 T cell response, which is illustrated in this slide, went over time in some elite controller while they're still maintaining a very good and excellent control of HIV infection, as shown on this slide, with a very reduced level of plasma viremia and the decrease of the response in the controller. So probably there is other response that have to be considered in the elite controllers as well. We have this very robust CD8 response, which is likely important for establishing the control of the infection in the controller. But as I said, this effector response maybe no longer required later on. The controllers are characterized by a very early control and an extremely low level of reservoir, and how they maintain this low level of reservoir, we have to take into consideration certainly other mechanisms, and we are trying to understand innate mechanism. Among innate mechanisms, the fact that the macrophages, as I said before, but also the CD4 T cells of the controllers have a reduced susceptibility of infection, which is illustrated on this slide, may have to be considered. And we have a very nice correlation indeed by the reduced susceptibility of infection in vitro, which is reported in this part of the slide, and the fact that the controllers have a very low level of reservoir. So we have to certainly understand better the mechanism of restriction of infection of these cells in the controller. Another thing that we have to consider is the NK cell response. We have recent data that are not yet published on phenotypic and functional feature of HIV NK cell activation in the controllers, which indicate that there is an activation of NK cells in the controllers, which might be different than the one in viremic patients. On this slide, you can see that we could not find out too much difference at the level of expression of CD69. However, and I'm sorry, but there is a problem with this slide. We found out that in the controller we have a higher level of expression of the NK cell receptor NK P44, which probably indicate an activation of NK cells. We also found out other markers that are upregulated in the controllers, like a marker which is related to an activation of ADCC in the elite controllers. So NK cell response probably has to be considered in the controllers as well. This slide, which is unfortunately not presented very well in the screen, was indicating that there is a good correlation between the CD8 response, suppressive response in the controllers, and indeed the NK activation in the controllers, looking for the expression of CD107, which is, as you know, a marker of cytotoxic capacity of NK cells. So in the controllers, we certainly have to consider both innate and adaptive response in the control of HIV infection. Is it the same situation in the post-treatment controllers? Certainly not. It's what the data of Azier in the lab is indicating. This slide, remember the main characteristic of the post-treatment controllers, we call it the Visconti patients. It's now 20 patients, more than 20 patients that we have in France, which has been treated very early on. They start the treatment within 10 weeks following the infection. They have been on treatment during 3 years, and they stop their treatment in agreement with their doctor. Now, for almost 10 years, they are off treatment, and they are doing perfectly well. As you can see on this slide, in opposite to the elite controller, they don't have the favorable MHC, HLA-B27, B57, but in opposite, in the post-treatment controller, we found the unfavorable MHC, B35, which indeed may correspond to the fact that most of the controllers, the post-treatment controllers that we have studied up to now, if they were treated so early, it's because they had a symptomatic primary infection with a high viral load at that time. However, the treatment was very efficient in the controllers and in the post-treatment controllers, and this treatment early on was not associated with a very strong suppressive CD8 immune response as we found in the elite controllers, as you can see on this slide. It's clearly associated to very low size of the reservoir, probably because of the very early treatment in those controllers. And as you can see also on this slide, there is in a number of controllers a decrease of the size of the reservoir over time, as nicely seen here in this part of the slide. So if we compare the elite controllers and the post-treatment controller, there is differences in the two situations. In the elite controllers, there are asymptomatic primary infection with a low viral load and a very high CD4 count during the primary infection. In the post-treatment controllers, as I said, they were symptomatic during the primary infection with high viral load, low CD4 count, and is the reason why they started the treatment very early on. As you see on this slide, around 80% of the elite controllers are the favorable HLA allele, HLA B27, B57, which is not the case at all for the post-treatment controllers. 5% of them, 57% of them are carrying the HLA B35, as I mentioned before. They don't have the strong CD8 response in the post-treatment controllers, as we have shown in the elite controllers. One very interesting point is the fact that I mentioned before that in the HIV controller, we have the CD8 cells that highly express HLA-DR, low expression of CD38 for the cells. In the post-treatment controller, most of them have very low T-cell activation. This is really one of the characteristics of the Visconti patients. The estimated frequency of HIV controllers is very low, 0.5% maximum of HIV-infected patients. For the post-treatment controllers, the estimated frequency is between 5 to 15% of people infected by HIV that can control the infection. So, if we look the study that has been made up to now on patients that are capable to control their infection, we can say that we have to consider both restriction of their cells to HIV infection. We have to consider the innate responses, both the NK response, which probably depend on the interaction with the plasmacytoid dendritic cells. We have to consider the priming by MDCs, as I mentioned before. And as a consequence, we have a reduction of the virus replication and the spreading of the virus in the reservoir, in the host, limiting the size of the reservoir and an optimal T-cell response, which is found in particular in the elite controllers. Can we learn from other models, like the primate models? It's a question that Michaela Müller in my lab is trying to understand now since years by studying non-pathogenic SIV infection in African monkeys. African monkeys have really special with characteristics that indicate high plasma viremia in the African monkeys, despite the fact that they do not develop the disease. When you compare pathogenic and non-pathogenic infection, and we have a summary on this slide, this is the only common feature between pathogenic infection and non-pathogenic infection, as you see on this slide, this high plasma viremia. But monkey in Africa, both African green monkeys and mangabeys, have a low viral load in their lymph node. This low viral load in their lymph node is associated with a lack of abnormal chronic T-cell activation that we found in pathogenic infection. And this attenuated T-cell activation is related to the fact that the monkey do not develop AIDS, both African green monkey and the sooty mangabey. So one hypothesis of the work of Michaela, but also of the work of others like Guido Silvestri in United States, is the fact that PDC, when they are exposed to HIV, produce, of course, the largest amount of type 1 interferon that will activate NK cells and MDCs, and as a consequence, the activated cells will produce cytokine, will migrate to the lymph node where T cells will be activated. What's happening in non-pathogenic infection? And Michaela and others has been very interested in looking for the level of production of cytokine early on after exposure to the virus in the monkey, in particular interested in study the expression and production of type 1 interferon. So this summarizes the data she obtained by comparing the production of type 1 interferon in the plasma of both African green monkey and pathogenic infection in macaque. As you can see on this slide, there is a lower production of type 1 interferon in AGM compared to pathogenic infection. We can see the same in the lymph node regarding the percentage of cells that are producing type 1 interferon in the lymph node comparing AGM situation and the macaque situation. Michaela has looked by microarrays with the expression in the lymph node, in the CD4 cells of the lymph node of interferon-stimulated genes, whether the profile was similar or not in both pathogenic and non-pathogenic infection. You can see on this slide that interferon-stimulated genes are expressed early on after the infection, but it's only a transient expression in the African green monkey compared to the pathogenic infection where the interferon-stimulated genes are persistently expressed. So the question that Michaela's group is asking, do we have any difference observed in type 1 interferon production which might be involved in the attenuated immune activation that we see in the African green monkey? And the question and the experiment that he did is what is happening if we increase type 1 interferon level during the acute phase of infection. And for that, of course, she made a study in the animal. First of all, she looked for the induction of early cytokine in African green monkey infected by SIV, and she found that by comparing again with the pathogenic infection, the main markers that we found in both models, but at a lower level for type 1 interferon and IL-15, are these cytokines. Of course, she wondered now what's happening if we inject type 1 interferon in the monkey during the acute phase of infection. What will be the outcome of the infection? What will be the outcome of different markers, including interferon-stimulator gene, including T-cell activation, viremia, CD4 cell count in the monkey, NK cell activation, dendritic cell markers and maturation, as well as inflammatory response after treatment by type 1 interferon. So she made that experiment and compared to untreated animal. And you can see on this slide that the data, both on the viral load, on the T cell count, on CD4 T cell proliferation and CD8 T cell proliferation in the monkey, indicate by that high dose of treatment by type 1 interferon during acute infection has no sustained effect on these different markers of disease progression in the monkey if you compare the blue and the black line on this slide. Now, what about impact on interferon stimulator gene? Nothing as well, as you can see here, no differences. No differences as well in terms of PDCs, as found, as you can see here. No consequences of the treatment by type 1 interferon on the number of NK cells as well. Just a slight difference in terms of NK cell activation after injection of type 1 interferon during the acute phase of infection. So we started to look at it a little bit more about NK cell response. And as you can see on this slide, we found out a stronger activation and proliferation of NK cells in the lymph node of the monkey, and a good correlation between the percentage of NK cells that proliferate and are activated in the lymph node of the monkey. And now, of course, what we are wondering is whether the NK cytotoxic activity might play a role in the control of HIV infection in the lymph node, since I said before that the main difference is, even if we have replication in the blood, we don't have a high viral load in the lymph node of the monkey. So this might play a role, and the conclusion of this study is that early cytokines are strongly induced during the acute phase of infection in the monkey, but the control of immune activation in the African green monkey seems to be triggered very early, and at least earlier than we previously considered. The African green monkey are still able to control immune activation even in the presence of very high level of type 1 interferon during acute phase of infection, and NK cells may play a role very early on in those monkeys. So I like to finish by saying that this kind of work, which is quite similar, I think, to the work that you are doing here at the Kirby Institute, is trying to integrate as much as possible basic science together to translational research with the objective to go to clinical research and clinical care. I think this is the objective of the CURE initiative that David mentioned before, that we launched at the International AIDS Society, to try to have more collaboration, more interaction between basic scientists and clinical researchers, but I don't forget also to integrate as well social science and interaction between communities and researchers. It is the way to go if we want to move forward towards an HIV cure in the future. So finally, I'd like to thank my collaborators, Michaela Müller and Azier Saessirians, and Daniel Scott Algara for the work on NK cells. This work is supported by the National Agency of AIDS Research and Viral Hepatitis C in France. We have the support as well of SIDAction. This work is part of the Vaccine Research Institute in France. We have recent support from AMFA as well for the work related to Visconti patient. I'd like to close there and to answer to your question.